Mycobacterium tuberculosis (MTB), also known as Koch's bacillus, is a species of pathogenic bacteria in the family Mycobacteriaceae, and the causative agent of tuberculosis. 1-2. First discovered in 1880 by Robert Koch. M. tuberculosis has an unusual, waxy coating on its cell surface primarily due to the presence of mycolic acid. This coating makes the cells impervious to gram staining, and as a result, M. tuberculosis can appear weakly gram-positive. 3. Acid-fast stains such as Ziehl-Neelsen or fluorescent stains, such as auramine, are used instead to identify M. tuberculosis with a microscope. The physiology of M. tuberculosis is highly aerobic and requires high levels of oxygen. Primarily a pathogen of the mammalian respiratory system, it infects the lungs. The most frequently used diagnostic methods for tuberculosis are the tuberculin skin test, acid-fast stain, culture, and polymerase chain reaction. In 2019, M. tuberculosis was found in a genetically related complex group of Mycobacterium species called Mycobacterium tuberculosis complex that has at least 9 members. M. tuberculosis 7 sensu stricto, M. africanum 7, M. canneti 7, M. bovis 7, M. capre 7, M. microti 7, M. pinnipedi 7, M. mungi 7, M. origus 7. It requires oxygen to grow and is non-motile. 8-9. M. tuberculosis divides every 18 to 24 hours. This is extremely slow compared with other bacteria, which tend to have division times measured in minutes. Escherichia coli can divide roughly every 20 minutes. It is a small bacillus that can withstand weak disinfectants and can survive in a dry state for weeks. Its unusual cell wall is rich in lipids such as mycolic acid and cord factor glycolipid, is likely responsible for its resistance to desiccation and is a key virulence factor. 10-11. Microscopy. Edit. Other bacteria are commonly identified with the microscope by staining them with gram stain. However, the mycolic acid in the cell wall of M. tuberculosis does not absorb the stain. Instead, acid-fast stains such as Ziehl-Neelsen stain or fluorescent stains such as auramine are used. 4. Cells are curved rod-shaped and are often seen wrapped together due to the presence of fatty acids in the cell wall that stick together. 12. This appearance is referred to as cording, like strands of cord that make up a rope. 9. M. tuberculosis is characterized in tissue by caseating granulomas, containing Langhans giant cells, which have a horseshoe pattern of nuclei. Humans are the only known reservoirs of M. tuberculosis. A misconception is that M. tuberculosis can be spread by shaking hands, making contact with toilet seats, sharing food or drink, or sharing toothbrushes. However, major spread is through air droplets originating from a person who has the disease, either coughing, sneezing, speaking, or singing. 17. When in the lungs. M. tuberculosis is phagocytosed by alveolar macrophages, but they are unable to kill and digest the bacterium. Within the M. tuberculosis genome, 3 generations of VNT or typing for M. tuberculosis are noted. The first scheme, called exact tandem repeat, used only 5 loci, 37, but the resolution afforded by these 5 loci was not as good as PFGE. The second scheme, called mycobacterial interspersed repetitive unit, had discrimination as good as PFGE. 38-39. The third generation mycobacterial interspersed repetitive unit 2 added a further 9 loci to bring the total to 24. This provides a degree of resolution greater than PFGE and is currently the standard for typing M. tuberculosis. 40. However, with regard to archaeological remains, additional evidence may be required because of possible contamination from related soil bacteria. 41. Antibiotic resistance in M. tuberculosis typically occurs due to either the accumulation of mutations in the genes targeted by the antibiotic or a change in titration of the drug. 42M. Tuberculosis is considered to be multi-drug resistant MDR-TB if it has developed drug resistance to both rifampicin and isoniazid, which are the most important antibiotics used in treatment. Additionally, extensively drug-resistant M. tuberculosis XDR-TB is characterized by resistance to both isoniazid and rifampin, plus any fluoroquinolone and at least one of 3 injectable second-line drugs, i.e. amikacin, kanamycin, or capreomycin. 43.
Measures is of utmost priority. An intrinsic contributor to the antibiotic-resistant nature of M. tuberculosis is its unique cell wall.
Saturated with long-
chain fatty acids or mycolic acids, the mycobacterial cell presents a robust, relatively insoluble barrier. 74. This has led to its synthesis being the target of many antibiotics, such as isoniazid. However, resistance has emerged to the majority of them. A novel, promising therapeutic target is mycobacterial membrane protein large 3-MMPL3, 75. The mycobacterial membrane protein large MMPL proteins are transmembrane proteins, which play a key role in the synthesis of the cell wall and the transport of the associated lipids. Of these, MMPL3 is essential, knockout of which has been shown to be bactericidal. 75 due to its essential nature, MMPL3 inhibitors show promise as alternative therapeutic measures in the age of antibiotic resistance. Inhibition of MMPL3 function showed an inability to transport trehalose monomycolate, an essential cell wall lipid, across the plasma membrane. 75. The recently reported structure of MMPL3 revealed resistance-conferring mutations to associate primarily with the transmembrane domain. 76. Although resistance to preclinical MMPL3 inhibitors has been detected, analysis of the widespread mutational landscape revealed a low level of environmental resistance. 76. This suggests that MMPL3 inhibitors currently undergoing clinical trials would face little resistance if made available. Additionally, the ability of many MMPL3 inhibitors to work synergistically with other anti-tubercular drugs presents a ray of hope in combating the TB crisis. Host genetics edit. The nature of the host pathogen interaction between humans and M. tuberculosis is considered to have a genetic component. A group of rare disorders called Mendelian susceptibility to mycobacterial diseases was observed in a subset of individuals with a genetic defect that results in increased susceptibility to mycobacterial infection. 77. Early case and twin studies have indicated that genetic components are important in host susceptibility to M. tuberculosis. Recent genome-wide association studies (GWAS) have identified 3 genetic risk loci, including at positions 11p13 and 18q11, 78-79. As is common in GWAS, the variants discovered have moderate effect sizes.
DNA repair, edit. As an intracellular pathogen, M. tuberculosis is exposed to a variety of DNA-damaging assaults, primarily from host-generated antimicrobial toxic radicals. Exposure to reactive oxygen species and/or reactive nitrogen species causes different types of DNA damage, including oxidation, depurination, methylation, and deamination that can give rise to single and double-strand breaks. DSBS. DNA depolymerase is upregulated in M. tuberculosis by several DNA-damaging agents, as well as during infection of mice. Loss of this DNA polymerase reduces the virulence of M. tuberculosis in mice. DNA2 is an error-prone DNA repair polymerase that appears to contribute to M. tuberculosis survival during infection. The 2 major pathways employed in repair of DSBS are homologous recombinational repair HR and non-homologous end joining in HEJ. Macrophage internalized M. tuberculosis is able to persist if either of these pathways is defective, but is attenuated when both pathways are defective. 81. This indicates that intracellular exposure of M. tuberculosis to reactive oxygen and/or reactive nitrogen species results in the formation of DSBS that are repaired by HR or in HEJ. 81. However, deficiency of DSB repair does not appear to impair M. tuberculosis virulence. In animal models, history of tuberculosis. M. tuberculosis, then known as the tubercle bacillus, was first described on the 24th of March 1880 by Robert Koch, who subsequently received the Nobel Prize in Physiology or Medicine for this discovery in 1905. The bacterium is also known as Koch's bacillus. Vaccine. The BCG vaccine botulinum calmette-guerin, which was derived from M. bovis, while effective against childhood and severe forms of tuberculosis, has limited success in preventing the most common form of the disease today, adult pulmonary tuberculosis. 86. Because of this, it is primarily used in high tuberculosis incidence regions and is not a recommended vaccine in the United States due to the low risk of infection. To receive this vaccine in the United States, an individual is required to go through a consultation process with an expert in M. tuberculosis and is only given to those who meet the specific criteria. The DNA vaccine, according to an article of the Journal of Preventive, Diagnostic and Treatment Strategies in Medicine September 2020, to DNA vaccine construct construct formation using Mycobacterium-specific gene in a specificities that DNA vaccine can be used alone or in combination with BCG. DNA vaccines have enough potential to be used with TB treatment and reduce the treatment time in future. Tuberculosis has existed throughout history, but the name has changed frequently over time. In 1720, though, the history of tuberculosis started to take shape into what is known of it today, as the physician Benjamin Martin described in his A Theory of Consumption. Tuberculosis may be caused by small living creatures transmitted through the air to other patients.